Hello everyone, welcome back to Infinity Learn by Sri Chaitanya. My name is Kiran Chaudhary and I am your botany faculty. I am back with another amazing session, my dear students, that is going to be extremely helpful for uplifting, for increasing your botany score in the coming NEET 2022. So let's start this session, my dear students. Let me tell you what are we going to do in this class. I am going to discuss with you 50 must-do questions from 3 chapters: Anatomy of Flowering Plant, Cell, the Unit of Life, and Cell Cycle and Cell Division. These 3 are very important chapters for your coming NEET 2022. A lot of questions are asked from these 3 chapters. So let us see what are the important questions that you must solve or practice before sitting in your NEET 2022 on 17th of July. So hello everyone, hello Neha, Aruna, and what's your name? Your name is Status. Can you please elaborate your name? Can you please let me know what exactly your name is? So as I say, before starting the class, if you are new to our channel, do consider subscribing to Infinity Learn NEET channel for more such videos and content in future. Now students, another important information that I want to share with everybody present in the class is, so I request you all, my dear students, okay, so today YouTube is not open, okay fine, so we'll get back to where I was explaining. Okay, so what exactly I was trying to explain over here is, my dear students, listen this very carefully. Wherever you are watching me right now on YouTube, what you should do is go into the description box where exactly you are seeing me, right? Where you have clicked and watching this video. Go into the description box and you can see several links over there, right? Okay, that's really nice. But may I know your name? Okay, your name is W. Your name is W, is that so? So go into the description box, my dear students, and there are several links provided to you. One of the first link that you will see over there is related to our, or that will take you to our website. So do consider visiting to our website and check out the amazing courses that we are offering there. So let's start the class now, and the topics that I will be covering in today's session are Anatomy of Flowering Plant, Cell, the Unit of Life, Cell Cycle and Cell Division. Okay, and I will really appreciate if you will participate in the session as well. So let's see the very first question. So again, the questions will be according to the chapter. So this is chapter wise. Okay, so the first chapter that I'm going to take is Anatomy of Flowering Plant. And here comes the very first question. Come on everybody, give it a try. Do not think whether your answer is going to be right or wrong. What I expect maximum from you in this class is try. Until and unless you try, my dear students, I won't be able to help you, right? So just focus on trying. So try to attempt the question. Phloem in gymnosperms lack. Phloem in gymnosperms lack. So can you tell me phloem in gymnosperms lack which of the cells? Yes, Aruna. Aruna says it is C. What about W? Neha? Okay, W thinks it's A. Okay, what about others? What about Neha? Neha, my dear, give it a try. Yes, come on, give it a try, Neha. See, Neha and other students, what I expect is try. Whosoever is listening but not trying, not participating in the session, I request you, please participate in the class as well. Come on, give it a try, my lovely kids. Give it a try. What do you think is the right answer? So W is actually correct over here. Phloem in gymnosperms will lack both sieve tubes and companion cell. So notice this, my dear students, presence of sieve tubes and companion cells is a characteristic feature of एंजियोस्पर्म्स, ये एंजियोस्पर्म्स का कैरेक्टर है, right? Whereas in gymnosperms, albuminous cells and sieve cells are present. So if somebody asks you what is present in gymnosperms, it is albuminous cells and sieve cells in their phloem. Right? Albuminous cells and sieve cells are present in gymnosperm. Is this clear, everybody in the class? Is this much clear? My lovely kids, let's go to the next question. Hello, Neha. Okay, the next question then. Regeneration of damaged growing grass which occurs due to grazing by the herbivores is largely due to. So which of the following meristem will help in regenerating the parts of the grass that are eaten up by the grazing animals? Come on, give it a try, my lovely kids. Everybody in the class, give it a try. कोशिश कीजिए आंसर देने का. Yes, everyone. What do you think is the right answer? W, Neha, Aruna, give it a try, my kids. Come on, trying is the maximum I expect you in this session. Okay, so the answer is actually intercalary meristem. So intercalary meristem is what? It's a meristem, it's a meristematic tissue that is present between mature tissues. You can say at the base of the internodes, or you can say at the base of the petioles, etc., where these intercalary meristem in grasses occur and regenerate the parts removed by the grazing herbivores. So it is the meristem that is present between the mature tissues. Is this clear? Let's take the next question. Meristematic tissue responsible for increasing the girth of the tree trunk. So what is the girth? Girth is the width, right? So which of the following meristem is responsible for increasing the width of the tree trunk? Can you tell me which one is this? Can you tell me which one is this? Hello Gyaneshwar, very warm welcome to this session, my dear. So everyone, give it a try. That's absolutely correct. The answer is going to be lateral meristem. Neha, Gyaneshwar, excellent. What about Aruna and W? You people have not participated in this question. Come on, every question is important for your exam. Exam is just around the corner. On 17th it is. You cannot afford skipping questions. You have to attempt it. If it is right, excellent. If not, then learn. This is the time to learn, right? Yes? Okay. So this is the lateral meristem which is present on the lateral sides of the plant and helps in increasing the width or girth of the tree. So let's take a little discussion over lateral meristem. They lie along the side of the stem. So they lie along the side of the stem, lateral side of the stem. They help in increasing the thickness of the plant, that is the girth of the plant. Examples of lateral meristem are vascular cambium and cork cambium. So vascular cambium and cork cambium help in increasing the width of the plant parts, be it a stem or a root. Next, vascular bundles in monocots monocotyledons are considered closed. So can you tell me why the vascular bundles are considered closed when it comes to monocot plants? Why the term closed is given? Come on, Gyaneshwar, Neha, W, everybody in the class. Aruna, Neha, come on. Come on, my lovely kids. Give it a try. Trying is maximum I expect you to do in this class. Yes, that's absolutely correct. Absence of cambium make these vascular bundle closed, and it is a characteristic of monocot plant. So monocot plants do not show secondary growth. Why they do not show secondary growth? Because in them cambium is absent. So let's revise a little about the vascular bundles. So these are the two categories of vascular bundles. One is open and another is closed. So let's discuss in length. So this is how an open vascular bundle look like, where it has a cambium strip, a layer of cells which are meristematic in nature between phloem and xylem. So between phloem and xylem there is a strip of cambium. Such type of vascular bundles are considered open because of the presence of this cambium. Okay, I should not make it with red color, right, because the xylem is already red in color. So I am drawing it with a green color. Next is, my dear students, so why it is open? Because cambium is present. Where can you see such kind of vascular bundles? You can find such kind of vascular bundles in dicot stem. They possess the ability to show secondary growth by forming secondary xylem and secondary phloem during secondary growth. So presence of cambium helps these dicot plants, especially these presence of open vascular bundles in dicot stem, help them to show secondary growth, formation of secondary xylem and phloem. Another type is closed vascular bundle. So if you see, the green color strip that was there in this open one is absent here. So there is no cambium here. Because of the absence of the cambium, my dear students, such vascular bundles are considered as closed. So there is no cambium between phloem and xylem. Such type of closed vascular bundles are found in monocot stem, right? Please notice this, my dear students, and also note one more thing. It's not one more thing that you need to understand over here is, when it comes to monocot plant, whether it is root or whether it is stem. Hello Virendra, good afternoon, very warm welcome in this session. So what you need to understand over here is, my dear students, in the entire monocot plant, whether it is root or stem, cambium is actually, or rather I should say, secondary growth is not found. But when it comes to the type of vascular bundles which is present in the stem and root of the monocot plant, so remember, in the roots generally radial type of vascular bundles is found, and these two vascular bundles that are displayed here are conjoint type. That means xylem and phloem present on the same radii, right? So presence of conjoint vascular bundle is a characteristic of stem. So conjoint open is found in dicot stem, conjoint close is found in monocot stem. That is why I have mentioned monocot stem here. Do not get confused. Okay, that's Shweta. Thank you very much. I forgot that. Thank you very much for reminding me. So again, they will not show secondary growth because of the absence of the cambium. Next. So another type of vascular bundles, the categorization is radial and conjoint, as I was talking about. So in radial vascular bundles, xylem and phloem are present on two different radii, right? So xylem and phloem in a vascular bundle are arranged on an alternate ring manner, right? They have different radii, and such type of radial vascular bundles are seen in roots. Conjoint means where xylem and phloem are present on the same radii, and if you see there is a green strip in them, that means they are open type. So xylem and phloem are jointly situated along the same radii of the vascular bundle. Such type of vascular bundle are considered conjoint, and they are commonly seen in stem and leaves. Do not forget, conjoint open in dicot stem, conjoint close in monocot stem. Is this much clear? Yes, my dear Neha, these questions will be posted in the Telegram. Everybody who's new to this class, please notice, join Telegram channel of Infinity Learn NEET. In the description box you can find the link. Okay, and this PDF of today's class will be shared over there. Next question, another amazing question. See over here, my lovely kids. A transverse section of plant show anatomical features. I repeat myself, a transverse section of a plant shows anatomical features like this. So you have to read out the four and find out these anatomical features belong to a monocot stem, dicot stem, or a monocot root or a dicot root. So large number of scattered vascular bundle. I think, my dear students, first statement itself is enough to find out that which type of a structure is this. Large number of scattered vascular bundle surrounded by bundle sheath. Large conspicuous parenchymatous ground tissue. Vascular bundles are conjoined and closed. When it has said vascular bundles are closed itself, another hint number 1 was this. Hint number 2 is they are monocots. Hint number 3, conjoined. So right now I have explained you conjoined where in stem and closed monocot stem, right? So conjoined in stem, but which stem? Whose stem? Monocot stem, because the vascular bundle is closed. So monocot stem is the right answer. Phloem parenchyma is also absent in monocot stem. That's absolutely correct. So please note this amazing question. That's absolutely correct, Aruna and Gyaneshwar. Next question. So let's revise in a little detail about monocot stem. This is how the anatomical structure of a monocot stem look like. This is how a monocot stem look like if you go and see it under a electron microscope. So let's talk about the characteristics of the vascular bundles that are found in a monocot stem. So vascular bundles are large in number and they are scattered, right? Large in number. What are these? These red portions. There are big red portions. There are big red portions in middle and smaller large portions on the periphery. So these red portions are nothing but the vascular bundle. They are large in number. These vascular bundles are surrounded by a sclerenchymatous bundle sheath. They are conjoined and closed, present on the same radii but absence of cambium. Peripheral vascular bundles which are present towards the periphery, they are small, and in the central they are large. Phloem parenchyma is absent and water containing cavities are present within the vascular bundles. Is this much clear, everyone? Is this much clear, everyone, my dear students? Let me know in the chat box. Is this much clear? Is this much clear? Chalo, let's see the next question then. In a dicot root, vascular cambium originates from. So can you tell me from where exactly vascular cambium originates when it is the case of a dicot root? We are not talking about stem. Be careful. Yes. Hello Savita. Very warm welcome in this class, my lovely kid. Shweta, Aruna, Savita, Gyaneshwar, Neha. Come on. Let me know what's the right answer to this question. Try everybody. W, yes. Yes, that's absolutely correct. So in a dicot root, tissue which is located below the phloem bundle and a part of protoxylem, and a part, my dear students, a part or a portion of pericycle tissue above the protoxylem, they participate in formation of vascular cambium. So see over here, a part, let me change the ink, portion which is below the phloem, a portion or a tissue located below the phloem bundles and a portion of pericycle above protoxylem. This is xylem, this is protoxylem, a portion above protoxylem. These are the tissues which participate in formation of vascular cambium in a dicot root. So in a dicot root, first of all, vascular cambium is completely secondary in origin. Entire vascular cambium will be formed during secondary growth. Tissues involved in formation of cambium ring are tissues located just below the phloem bundles and a portion of pericycle tissue above the protoxylem. Is this much clear, everybody? Any doubts till now? Now when it comes to the vascular cambium in a dicot stem, it is mixed in origin. It is primary as well as secondary. So if you remember, vascular cambium in a dicot stem, it is made up of interfascicular and intrafascicular, right? So intrafascicular was formed during the primary growth itself, right? And then during secondary growth, the medullary rays that is present between the intrafascicular cambium, they will become meristematic by undergoing the process of de-differentiation, and they will lead to a formation of another cambium cells known as interfascicular cambium. So interfascicular during secondary growth it was formed, intra was formed during the primary. So the vascular cambium in a dicot stem is mixed in origin, but in a dicot root it is completely secondary in origin. Everybody, is this much clear? Any doubts till now? Any doubts till now? Okay, excellent. Now let's take the next question. Identify the incorrect statement. Identify the incorrect statement. So come on, let me know which of the following is an incorrect statement. Everybody, let me know which of the following is an incorrect statement. Come on, give it a try. Everybody present in the class, give it a try. Neha, W, Gyaneshwar, Savita, Shweta, Aruna. Come on guys, give it a try. Identify the incorrect statement. Identify the incorrect statement. Come on. Okay, let me try this. Hardwood does not conduct water but gives mechanical strength is right. Sapwood is involved in conduction of water and minerals, right? Sapwood is the innermost. No, sapwood is the outermost. Actually, heartwood is the innermost. Heartwood is the innermost, hence this is the incorrect statement. Due to deposition of tannin, resin, oils, etc., hardwood is dark in color. That's right. Is this question clear? Excellent. Very nice, Gyaneshwar. Let's see the next question then. Which of the following statement given is not true about the formation of annual rings? Remember, annual rings help in determining the age of a tree. So by counting the annual rings, you can count the age of a tree. Which of the following statement is not true about formation of annual rings? Come on. Gyaneshwar, Neha, Aruna, Shweta, Neha, W. Come on. कोशिश कीजिए. कोशिश ही है जो मैं आपसे एक्सपेक्ट कर रही हूँ इस सेशन में. I don't expect you to always give right answer. I'm expecting you to just try. Forget about whether your answer is going to be right or wrong. Just try. I assure you, you won't make this same mistake again if you do right now. But for that, you need to at least try. Okay, so Aruna thinks it's A. Gyaneshwar thinks it's C. What about Neha, W? Savita? Okay, देखो. Okay, don't be angry. W. Fine. Read the statements. Okay, so now W, have you read the statements? What do you think is the right answer? Yes, W, what do you think is the right answer? Okay, now let me see. So you were supposed to find out the incorrect statement. Which of the following is not true? इसमें से गलत कौन सा है? Which of the following is wrong? You were supposed to find out. Annual rings are not prominent in trees of temperate region. This is in fact wrong, and this becomes the answer of this question. Why? Not prominent मतलब अच्छे से नहीं दिखेगा. Not won't be clearly visible, but that's wrong. In temperate region, there's a clear distinction between the seasons. Autumn की तरह दिखेगा, winter की तरह दिखेगा, spring की तरह दिखेगा, summer की तरह दिखेगा. That means there are clear variations, distinctions in the seasons, and you know these annual rings depends upon the season because the activity of the cambium is seasonal. It changes with the seasons. But if we compare it with a tropical forest or a tropical region, in tropics the climate does not varies or does not changes much. Hence there is no such not much difference between the seasons. Hence these annual rings cannot be formed so accurately or won't be that clearly visible or distinctly visible in a tree growing in a tropic. But in comparison to a tree that is growing in a temperate, these annual rings will be quite clear and distinct. So first one is incorrect. Did you get that, everybody in the class? Aruna, W, Shweta was correct over here. Gyaneshwar, do you know why? Is this question clear now? Annual rings is a combination of spring wood and autumn wood. Yes, spring or autumn clearly दिखता है temperate में. That is why these annual rings can be easily formed and will be clearly visible. Differential activity of cambium causes light and dark band of tissue, early and late wood. That's right. Activity of cambium depends upon the variation in climate, and hence the formation of annual rings takes place. Is this clear, everybody? Is this clear, everybody? Any doubts in this class? Any doubts in this question? Come on, please express yourself in the chat section. Okay, then get ready for the next question. Which of the following is made up of dead cells? Can you tell me which of the following is made up of dead cells out of these four? Yes, W, Neha, Gyaneshwar, Savita, Shweta, Neha, Aruna. Come on, my lovely kids, give it a try. कोशिश कीजिए. Give it a try. Which of the following is made up of dead cells? Yeah, that's absolutely correct. Phylum. देखो, xylem, parenchyma are the only component of the xylem which are living. ये only component है xylem के which will be living. Rest all components are dead. So this is not the correct answer. Phylum. What is phylum? Let me explain you, my dear students, with this simple diagrammatic representation. Phylogen, also known as cork cambium. So remember I told you when I was explaining you about secondary meristem, or rather I should say lateral meristem. When I was explaining you lateral meristem, I told you there are two examples of lateral meristem, that is vascular cambium and cork cambium, that will help in secondary growth. So cork cambium also helps in secondary growth. It also helps in increasing the girth of the plant parts. Another name for cork cambium is phylogen. That means phylogen is meristematic in nature. Now this phylogen will form new cells in both the direction. Few cells it will make, or rather I should say the cells that will be produced towards the center of the plant, they will differentiate into secondary cortex, right? And they will be given a specific term, that is phyloderm. The cells that are formed from cork cambium and which are towards the periphery, they will have a deposition of suberin in them, and suberin deposition is going to make the cell dead, and they will be considered as cork cells, and they will get deposited, or they will form the peripheral part of the plant, right? And they will be called as phylum. So phylum is known, another name for cork, which is getting deposited with the help of suberin, and because of which they become dead. So they are actually dead cells. What are dead cells? Phylum. Phylogen is cork cambium, phyloderm is the secondary cortex. So together phylum, phylogen and phyloderm is together known as periderm. Is this much clear, everybody in the class? Any doubts till now? Next question. Interfascicular cambium develops from the cells of. Interfascicular cambium develops from the cells of. Come on, everybody. Interfascicular cambium develops from the cells of. Everyone in the class. Interfascicular cambium develops from the cells of. Yes, give it a try. Yeah, that's absolutely correct. Medullary rays that are present between intrafascicular cambium. Medullary rays that are present between the intrafascicular cambium. These medullary rays between the intrafascicular cambium, these are the intrafascicular cambium present in the conjoint vascular bundle, right, in a dicot stem. So these are the medullary rays. They will undergo de-differentiation, where they will lose their ability to perform a specific function and regain an ability to divide. They will form these interfascicular cambium. Now together inter and intrafascicular cambium will make a vascular cambium ring, and they will participate in secondary growth, forming secondary phloem towards the periphery and secondary xylem towards the center. Excellent. Next question. Match the following and choose the correct option from below. Match the following and choose the correct option from below. Everybody, give it a try. Match the following and choose the correct option from below. Everyone, give it a try. Yes, give it a try. Aruna, Neha, Shweta, W, Gyaneshwar, Aruna, Savita. Please try this question. Okay, then let me help you out. Cuticle is a waxy layer, so A goes with 3. Bulliform cells are the empty colorless cells. I will explain you the function as well. Stomata, in them guard cells are present, so C goes with 1. And D, epidermis. Epidermis is single layer thick, single layer of the present in the plant parts. So A goes with 3, B goes with 4, C goes with 1 and D goes with 2. A is absolutely correct. A is absolutely correct. Very good. W, Shweta, Aruna, Princey. Very good. Next question. Okay. So what is a bulliform cell? First of all, you need to understand bulliform cell is a characteristic of a monocot leaf. Where they are present? They are present on the adaxial surface, this surface. They are present on the adaxial surface, this surface. These are the bulliform cells. They are present in the grass. Grass is an example of a monocot. Bulliform cells are a part of epidermal cells, right? So in grasses, certain adaxials, epidermal cell along the veins, what they will do, they will modify themselves into large, empty, colorless cells. Now what is the function of these cells? These cells, my dear students, has the ability to absorb and store water in them. So whenever there will be water available, they will store and make the leaf surface exposed to the environment. Whenever there will be scarcity of the water, they will lose the water, right, and makes the leaf surface curl inward so that they can reduce the surface area of the water, hence reduces the rate of transpiration, which is required when there is less water in the surroundings. Ma'am, screen is blur. Are you sure screen is blur? Because from my... Okay, just give me a moment. Is the screen visible now? So students are complaining the screen is not visible. Can you check once the internet connection? So what about for other kids? Princey, is it visible for you? Is it visible now? Yes, okay, so we'll continue then. Next question. Match the following and choose the correct option from below. So column 1 and column 2. So here there are different types of tissues present and here their function is present or maybe a structure related to it. So tell me, my dear students, this is a good question to revise all the important simple tissues, right? Simple tissues, be it meristematic or permanent. Let us try to revise them. Come on everybody. Come on everybody. Give it a try. Give it a try. So W says it's B, B, B. Let me check out. Meristem. A will go with actively dividing cell. Let me see. B, parenchyma support. Sclerenchyma can also provide. Right? But now in that case, my dear students, you need to see what are the options available. So another option available in the column 2 is sclerids. So sclerids can go with the sclerenchyma. So in such questions where the same option can match with two given options, my dear students, or in match the columns when one column can match to two columns in the second one, two options in the second column, in that case you need to go with what could be the most accurate or probable answer. So collenchyma we will keep with mechanical support, right, and sclerenchyma we will keep with sclerid. But do not get confused, sclerenchyma also provide mechanical support. In fact, they are the major tissues that provide the mechanical support, but since sclerids are mentioned, so we will keep sclerenchyma with the sclerids. Now E is the epidermis, so the answer is going to be epidermal tissues have stomata. Stomata is the modification of epidermal tissue. So it should be A3, B1, C2, D5 and E4. Excellent. The answer is B. Excellent. W. Aruna. Now let's see the next question. Epiblemma of root is equivalent to. So to which structure inside a root epiblemma term is valid for? So in which of the following layers present in a root you can give the term epiblemma? Come on everybody. Aruna, W, Princey. Come on. Shweta, Neha. Give it a try. What do you think is the right answer, kids? What do you think is the right answer? Come on, give it a try. I think it... No, you don't have to be sure or unsure. There's nothing like that, W. This is a practice session. Just try. So epiblemma of root is equivalent to epidermis. So epiblemma is a special term given to the epidermis of the root. Please remember this from now. Next question. Interfascicular cambium and cork cambium are formed due to. Interfascicular cambium and cork. Remember, it is talking about inter, not intra. Interfascicular cambium and cork cambium are formed due to. Come on, everybody. That's a good question. Aruna and Shweta thinks it should be what? It should be C. This is for this question or for the earlier one? Give it a try, everybody. Give it a try, everybody. So Shweta thinks this time it is B. Okay. What about Aruna? Aruna also thinks B. What about other kids? Come on. Everything is thinking B, but B is not the right answer. The right answer is D, differentiation. See students, whenever a meristematic cell converts into a permanent cell, what is the meaning of converting into a permanent cell? That means inducing of a particular function. So whenever a cell starts performing a particular function, it is known as a permanent cell. That means now it will start performing a particular function. It will no longer divide. So this process of meristematic cell converting into a permanent cell and starting to perform a specific function, this is known as differentiation. इसे कहते हैं differentiation. इसे कहते हैं differentiation. Now, sometimes what happens, these permanent tissue, they lose their ability to perform a specific function and regain their ability to divide again. This is known as de-differentiation. So, for example, I will talk about medullary rays. Medullary rays are the permanent cells that were present between the intrafascicular cambium in a dicot stem. These medullary rays are examples of permanent cell or tissues during secondary growth, they will undergo dedifferentiation. They will undergo dedifferentiation. Differentiation. So dedifferentiation क्या होता है? No, losing the ability. What is differentiation? Gaining an ability to perform a specific function. What is dedifferentiation? Losing again the ability to perform a specific function and gaining again the ability to divide. So इससे क्या बनेगा? It will lead to a formation of interfascicular cambium. Interfascicular cambium. So interfascicular cambium is formed. Interfascicular cambium is formed because of dedifferentiation happening in the medullary rays. So W, is this okay for you? Is this satisfying for you? Next, similarly, cork cambium cells are also produced because of dedifferentiation in the cortex cells present in the plant part, present in the dicot stem, right? So what happens, my dear students? They undergo in a dicot plant, the cortex region, in the cortex region, what will happen? Cortical cells will undergo dedifferentiation, will become meristematic and do the formation of cork cambium. These cork cambium will form secondary cortical cells towards the periphery and they will form cork cells of phylum towards the periphery. Is this okay now? Is this concept clear? So please remember this. Next question. A piece of wood. Now this is a tricky question. Let me see who can crack this one. Let me see who can crack this one. This is a little tricky question. A piece of wood having no vessels must belong to. A piece of wood having no vessels belong to. A piece of wood having no vessels belongs to. A piece of wood having no vessels belong to. A piece of wood having no vessels belong to. Come on everybody. A piece of wood having no vessels belongs to. Yes, that's quite impressive. Shweta, Princey, that's quite impressive. The answer is pine because pine is a gymnosperm and in gymnosperms vessels are not found. Presence of vessels is a characteristic of angiosperms like teak, mango and palm. They are examples of angiosperm. In gymnosperms like pine, vessels are not found. Is this question clear to everybody? Any doubts till now? Any doubts? Aruna, Shweta, Savita, Princey, W? Pine is a gymnospermic plant. Pine is a gymnospermic plant. Pine is a gymnospermic plant, my dear. If you do not know, please note from now. Next. Casparian strip occur in. Come on everybody. Casparian strip occur in. Casparian strips occur in. Casparian strips are made up of suberin. Casparian strips occur in. Come on. Give it a try. What do you think Casparian strips occur in? Which type of layer present in roots? Where, in which of the layer of the root you can find Casparian strip? Come on everyone, give it a try. Casparian strips occur in. Casparian strips occur in. Come on. Casparian strips occur in endodermis. They are present in endodermis of the root. Remember, that is why even in chapter number 11, transport in plant, remember, because of the presence of Casparian strips in the endodermal cells, the water needs to enter into the endodermal cells via symplast only. Apoplast is not possible in the endodermal cells. Excellent. Come on. Please remember this. This information is twice given in NCERT, one in chapter number 6 anatomy and then chapter number 11 transport in plant, where movement of water is discussed, long distance transport is discussed, absorption of water in plants is discussed. Is this clear? Next. Root hair develops from the region of. So what do you think, from which region of the roots root hair will develop? What do you think, from which region of the root hair will develop? What do you think, from which region of the root hair will develop? Come on, from which region of the root hair will develop? Come on everybody, that's absolutely right. They will originate from the maturation region. Please note this, that's quite simple but yet tricky. If you do not pay attention, you might end up giving the answer maybe meristematic active region, but that's not right. Remember, they comes out from the maturation zone. Next. So these were the questions, my dear students, that were absolutely important from your anatomy of flowering plant chapter 6 class 11. These were the must questions that you should practice before sitting into NEET 2022 on 17th July. Right? They help you in revising all the concepts, important concepts on which questions have already been asked or might be asked again, because NT has the history of repeating questions, right? खत्म, सिर्फ anatomy खत्म हुआ है. Now we will do cell the unit of life, then cell division, right? So let me know in the chat box, are you guys ready to practice questions from cell the unit of life? If you are ready, type ready in the chat box. Come on, type ready in the chat box. If you are ready, type ready in the chat box. Only Shweta and W are ready. What about the other kids? Aruna, Princey, Shweta, Neha. Okay, so let's start now from cell the unit of life. So here comes the very first question. Which structure performs the function of mitochondria in bacteria? Which structure performs the function of mitochondria in bacteria? Come on everybody, give it a try. Come on everybody, give it a try, my lovely kids. Give it a try. Come on everyone, give it a try. Come on everybody, give it a try. That's absolutely correct. The answer is mesosome. So mesosomes are the extensions. They are the extensions of plasma membrane, right? Plasma membrane in a prokaryotes like bacteria. So remember, bacteria is a prokaryote, and in prokaryote, membrane bound organelles like mitochondria will not be found. So who will perform the function of mitochondria? Who will help in cellular respiration? Who will help? Mesosomes will help. They are nothing but the extensions of plasma membrane. There are other several functions of the mesosomes as well. Hello Shakar, very warm welcome in this class. Next question. The term glycocalyx is used for, so can you tell me which is the right option that is valid for the term glycocalyx? Remember, glycocalyx word is used for a prokaryotic cell. Come on, Aruna. Shakar, W, Shweta, Neha, Princy, what do you think is the right answer? So, glycocalyx is used for a layer surrounding the cell wall of bacteria. So, in a bacteria, my dear students, prokaryote, example bacteria have a cell envelope. They possess a cell envelope, right? This cell envelope is made up of 3 layers. It is made up of 3 layers. The outermost is the glycocalyx. Inner to the glycocalyx is the cell wall made up of peptidoglycans, remember? And the inner to that is the cell membrane, right? So, these are the 3 structures, right? These are the 3 layers, right? Compactly arranged out of which the cell envelope of the bacteria cell is made up of. Now, this glycocalyx, according to this option, glycocalyx is used for a layer surrounding the cell wall. So, yes, the cell membrane is the innermost, glycocalyx is the outermost. So, technically glycocalyx is covering the cell wall. They have all 3 layers have their distinct specific roles, but togetherly they work in protecting the bacterial cells. So, they are protective in nature cumulatively. Is this clear, everybody? Glycocalyx further can be of 2 types depending upon the thickness and composition, slime layer and capsule. Is this clear? Any doubts till now? Is this question clear? W, Shakar, is this question clear? Yes, my lovely kids, is this question clear? Is this question clear? Is this question clear? Okay, so let's see the next question then. Which of the following statement is not correct? So, you are supposed to find out the incorrect statement. So, can you tell me which of the following statement is not correct? Can you tell me which of the following statement is not correct? Can you tell me which of the following statement is not correct? Can you tell me which of the following statement is not correct? Yes, come on. Let me help you out then. Lysosomes are formed by the process of packaging in endoplasmic reticulum. No, my dear students, this is directly, in fact, one of the first line given under lysosomes topic or heading, right? So, it is clearly mentioned that lysosomes are formed by the process of packaging in Golgi complex, not endoplasmic reticulum. Function of packaging is of Golgi complex and not endoplasmic reticulum. Right? So, this one is incorrect. Lysosomes have numerous hydrolytic enzymes, that's true, that is why they are digesting molecules or components of the cell. The hydrolytic enzymes are active under acidic pH, that is also right. Lysosomes are membrane bound structure, they are single membrane bound structure, this is also right. A is the right answer to this, not right option to this. I would say right answer hai ye A. But actually the statement is wrong and they are asking about the wrong statement. So hence A become the answer. But A statement is incorrect statement. Please remember this, the difference. Is this okay, W? Is this okay? Is this okay, my dear students? Can we move to the next question then? Can we move to the next question then? So again, match the following, match column 1 with column 2. Let me see who can match this correctly. Let me see who can match this correctly. Come on, everybody. Let me see who can match this correctly. Yes, Shakar, W, Aruna, Neha, Princy. You guys have stopped giving the answers. Come on, do not lose the momentum. Try कीजिए. Try to give the answer, please. So let me help you out here. Golgi apparatus. What is the function? Formation of glycoprotein and glycolipids. That means, my dear students, modification of the proteins and lipids. This is also one of the function of Golgi apparatus. Lysosomes have several digesting enzymes. Vacuoles, they trap the waste and excretory products. Ribosomes helps in synthesis of the protein. So A goes with 3, B goes with 4, C goes with 2 and D goes with 1. So A is the right answer. Excellent. W. Aruna. Let's see the next question, my lovely kids. What is the next question? The next question is about the nucleolus. Which of the following is correct or true about nucleolus, which is a structure found inside nucleus? Yes, come on. Come on, everybody. Give it a try, my kids. Give it a try, my lovely kids. Come on. Yes, what is the right answer? Large nucleoli are present. Acha, no, this is not the right one. Nucleolus is a site for active ribosomal RNA synthesis. So here large number of rRNA molecules are synthesized inside nucleolus. This is a straight NCERT based question. D is the correct answer. Is this clear now? Aruna, W, is this correct? Is this okay? Yes, that's right. Now, yes, so is this question clear? Shweta, W, Aruna, is this question clear now, my lovely kids? Is this question clear? Is this question clear? Come on, everyone. Is this question clear for everybody or not? Yes, so let's take the next question then. Mitochondria and chloroplast. There are two statements given. Mitochondria and chloroplast are semi-autonomous organelles. Yes, they are semi-autonomous organelles. There are two words over here. Autonomous के आगे क्या लिखा है? Semi. That means they are not completely autonomous. Right? What do I mean by completely autonomous? That means they do not need or they do not require help from any other part of the cell. But they are semi-autonomous. Why they are semi-autonomous? Because although they possess their own DNA, RNA and protein making machinery, but they are incapable of producing all the types of proteins they require for proper functioning. Most of the proteins that they require come from the DNA that is present, or they are formed with the help of transcription mechanism that is taking place with the help of DNA which is present inside the nucleus, right? So, how are proteins produced? First of all, mRNA will be produced from the DNA, then from mRNA, that mRNA will participate in the translation mechanism. So, most of the proteins they require come from the DNA that is there in the nucleus, hence they are semi-autonomous. So, both of them possess DNA molecule. They are formed by division of pre-existing organelles and they contain DNA but they lack proteins, and this is wrong. They do divide by fission, my dear students. They do contain DNA but they do not lack protein synthesizing machinery. So, this much portion is wrong, rest the statement is okay. They do divide via fission, that means from a pre-existing cell organelle, because remember mitochondria and chloroplast, they were once bacterial cells, right. So, mitochondria was probable and was an aerobic bacteria, and chloroplast was an autotrophic bacteria, photosynthetic autotrophic bacteria. It was engulfed by the eukaryotic cell during the course of evolution when the eukaryotic cells were evolving on earth. That is why they have this nature. That is why they possess their own DNA, which is actually double-stranded and circular, and possess the ribosomes, which are of 70S type. Is this clear? So first statement is correct, but B is false. Is this question clear to everybody? Let me know in the chat box. Everybody present in the class, if this much is clear, do let me know in the chat box. बेटा आपका और हमारा जो ये कनेक्शन है वो थ्रू चैट बॉक्स है. अगर आप मुझे चैट बॉक्स में नहीं बताओगे कि हाँ ये क्वेश्चन क्लियर है मैम, हमें दूसरा क्वेश्चन सॉल्व करना चाहिए, या इस क्वेश्चन में हमें ये प्रॉब्लम है, मुझे ये जानना है, या इस चीज़ के बारे में बताओ, हाउ विल आई हेल्प यू? सो आई कैन हेल्प यू ओनली एंड ओनली इफ यू गोइंग टू आस्क फॉर द हेल्प. मुझे कैसे पता चलेगा आपको कितनी मदद की और कहाँ पे मदद की ज़रूरत है, राइट? सो प्लीज लेट मी नो इन द चैट बॉक्स. बी एक्टिव इन द चैट बॉक्स और बताओ आगे बढ़ें या रुकें या सॉल्व करें. आप लोग बोलते ही नहीं हो, चुपचाप बैठे रहते हो. मुझे कैसे पता चलेगा कि वी कैन गो टू द नेक्स्ट क्वेश्चन और नॉट? बोलोगे तब तो पता चलेगा. यस, सो कोई भी चुप नहीं रहेगा. चैट बॉक्स में लिखते रहो, आगे जाना है, रुकना है, आंसर दो, गलत हो तो भी दो, राइट? उसी तो सीखोगे सही क्या है. जब गलत का पता होगा तभी तो आपको पता चलेगा कि सही क्या है. Now you people are making me talk too much, but what to do? You are not active. Okay, let's see the next question then. Major site for synthesis of lipids. So lipids सबसे ज़्यादा कहाँ पे बनते हैं, या लिपिड बनने की मेजर साइट का क्या है? A major site for the synthesis of lipid is, everybody, come on. Where do you think lipids are synthesized? Come on everybody. Nobody is going to be quiet and silent in the chat box. Try करो इस क्वेश्चन को. Try करोगे तभी तो आगे आंसर समझ में आएगा और एग्जाम में सही आंसर लिख पाओगे. Come on, try. Yes, that's right. Lipids are synthesized in the smooth endoplasmic reticulum, and rough endoplasmic reticulum is majorly considered or majorly concerned with protein synthesis. So SER, that is a smooth endoplasmic reticulum, it is devoid of ribosome on its surface. Rough endoplasmic reticulum के surface पे ribosomes हैं, right? So this SER is majorly concerned with the synthesis of lipids or fats. Next question. Which of the following statement about inclusion bodies is incorrect? Come on everybody. Which of the following statement about inclusion bodies is incorrect? अच्छा, ये inclusion bodies है क्या? Inclusion bodies are present. They are the characteristic feature. They are the characteristic feature of prokaryotes. तो prokaryotes का है ये feature. Prokaryotes का feature है ये. So tell me which of the following statement is incorrect about inclusion bodies. Come on everybody. W, Shweta, Aruna, Neha, Princy. Come on everybody. Shakar, come on. Don't be a silent spectator. Participate in the session. Try to give the answer. 17th को कैसे पुश करोगे अपने आप को आंसर देने में? Come on, you have to push yourself, your mind, right? ये तो अभी तो हमारा सेशन around 1 hour का हुआ है, right? Roughly it is 1 hour and 4 minutes. You have to sit there for 3 hours, right? 3 घंटे कैसे अपने दिमाग चलाओगे अगर ये घंटे भी नहीं चला पाओगे तो? And it's just biology right now. There you are supposed to answer botany, zoology, chemistry, physics, right? So you have to develop this stamina of making your brains, your neuron, giving that stress, that condition that yes, हमें 3 घंटे तक लगातार पेपर सॉल्व करना. We have to attempt the question. You just can't give up in just an hour, my kids. It is biology right now. तो इतना जल्दी तो आप लोगों को थकना है ही नहीं, right? Next. So इसमें right answer क्या है? I mean to say इसमें से इन 4 में से incorrect statement क्या है? So inclusion bodies are not bound by any membrane. That's absolutely correct. दे आर इन्वॉल्व इन इंजेक्शन ऑफ फूड पार्टिकल, नो नो नो, इनका कोई इंजेक्शन या डाइजेशन से कोई रोल नहीं है. दे रिप्रेजेंट द रिजर्व मटीरियल, राइट. So this is in fact wrong. They lie freely in the cytoplasm, that's right. They represent reserve material in the cytoplasm, this is also right. Clear everybody? That's good. Next question. Which of the following statement regarding mitochondria is incorrect? Which of the following statement regarding mitochondria is incorrect? Come on. Come on everybody, give it a try. Come on everybody, give it a try. बताइए इसका क्या आंसर होना चाहिए? Don't be silent, participate. Come on. एटलीस्ट आंसर तो दो चैट में, और कुछ बोलो या नहीं बोलो. आप लोग तो बिल्कुल ही साइलेंट हो गए. किड्स कम ऑन. ऐसा लग रहा है मुझे ही 17 को एग्जाम देना है ना. मैम ही ट्राई किए जा रही हैं. इट्स यू हैव टू पार्टिसिपेट. 17 तारीख को एग्जाम आपका है बेटा, आपका लाइफ डिसाइड होगा. मेरा तो हो गया जो होना था, एटलीस्ट 17 तारीख से तो नहीं होगा. So let me read out this. Which of the following statements is incorrect? ठीक है. Mitochondrial matrix contain single circular DNA molecule and ribosomes. ये सही है, ध्यान रखना, सिंगल सर्कुलर होता है. और plastids में, right, there can be many DNA molecules. This is NCERT. This is how NCERT writes. DNA molecules लिखा है plastids में. ये क्लोरोप्लास्ट भी कह सकते हैं. Outer membrane is permeable to monomers of carbohydrate, fats and proteins. So outer membrane is more permeable in comparison to inner membrane. Remember, mitochondria is a double membrane cell organelle. Enzymes of electron transport are embedded in outer membrane. That's wrong. ETS के जो enzymes हैं वो क्या आउटर मेम्ब्रेन में है? ETS के जो कॉम्प्लेक्सेस हैं वो क्या आउटर मेम्ब्रेन में? नो, दे आर प्रेजेंट इन इनर माइटोकॉन्ड्रियल मेम्ब्रेन. ETS runs in inner mitochondrial membrane. Inner membrane enfolds to form crista. That's absolutely correct. That's absolutely correct. So this one is wrong. Let's see the next question. Which of the following cell organelles is enclosed by a single membrane? So इनमें से ऐसा कौन सा ऑप्शन है या इनमें से ऐसा कौन सा सेल ऑर्गेनल है जो सिर्फ सिंगल मेम्ब्रेन से कवर्ड है? That means rest 3 are covered by 2 membranes. Yes, it's a little tricky question. Let me see who can crack this one. Come on, Aruna, Shweta, W, Neha. Come on, give it a try, my lovely kids. कोशिश करो. Give it a try, my lovely kids. Come on, कोशिश कीजिए. Yes, Princey, that's absolutely right. Lysosomes is covered by a single membrane. Nucleus, mitochondria, chloroplast, all are covered by double membranes. Very important, remember this. Next, match. Match column 1 with column 2. Let me see who can match correctly. Yes, let me see who can match correctly. Come on, everybody. Let me see who can match correctly. Come on, come on, come on, be quick. Be quick, my lovely kids. Let me see then. Thylakoids. They are the flat sac-like structure in the stroma. Cristae is the infoldings of the inner mitochondrial membrane. Next, cisternae are disc-shaped sac in Golgi apparatus. Chromatin is the condensed structure of DNA. So A goes with 3, B goes with 4, C goes with 1 and D goes with 2. That's absolutely correct. Excellent. It will be D. So is this question clear now? Shweta, is this question clear? Next, which of the following types of plastid does not contain stored food material? These are the types of plastids. Out of this, which of the following does not store any type of food material? Come on. Which of the following does not store any type of food material? Come on, give it a try. कोशिश करो, बताओ इसका क्या आंसर होना चाहिए? बताइए इसका क्या आंसर होना चाहिए? Come on, everybody. That's absolutely correct. Chromoplast has pigments that produces or that gives color to a particular plant organ. Elaioplast, aleuroplast and amyloplast. This is going to store starch or carbohydrates. Aleuroplast is going to store proteins. Elaioplast is going to store the fats and oils. Right? Next question. So answer is A is not going, chromoplast is not going to store any food. A common characteristic feature of a plant sieve tube and most of the mammalian erythrocyte. So mammalian RBC और plant में जो sieve tubes होती है उनमें क्या common है? What is common between a mammalian RBC and a sieve tube? Remember, we are talking about mature mammalian RBC and a mature sieve tube. में common क्या है? Come on, दोनों में common बताना है. Common क्या है मेरे प्यारे बच्चों? Common क्या है इनमें? Yes, common क्या है? Excellent. Absence of nucleus. Nucleus नहीं होता है दोनों में at the time of maturity. That's excellent, Shweta. Next question. What is tonoplast? ये tonoplast term हम किसके लिए use करते हैं? To what structure this term tonoplast is used? Come on. What is tonoplast? Everybody? Aruna, W, Shweta, Neha, Princey. Give it a try, my kids. That is absolutely correct. Membrane boundary of vacuole is termed as tonoplast. This is one of the, along with the tonoplast, plasma membrane along with the tonoplast helps in deciding the permeability of the substances as well. How many questions are present? Beta 50 questions हैं. इन पे क्वेश्चन नंबर इसलिए नहीं लिखा है ना ताकि आप लोग डिस्करेज ना हो कि अभी और कितने क्वेश्चन हैं. पार्टिसिपेट करो. सेल इज गोइंग टू फिनिश नाउ. W. सेल इज गोइंग टू फिनिश नाउ इन फ्यू मोर क्वेश्चंस. 2-3 क्वेश्चंस और होंगे. Then we will start with cell division. Next question. Select one which is not true for ribosome. Select which is not true for ribosome. Come on, everybody. Come on. We have covered more than half of the questions. Don't worry, W. You have to sit for 3 hours in NEET exam. Don't forget that. अभी तो 1.5 घंटा ही नहीं हुआ. अभी तो it's not just 1.5 hour is also not completed till now. So you can't lose energy and momentum. So which is not true? Yeah, this is wrong. Ribosomes play a role in protein synthesis. Ribosomes work as a protein factory. Ribosome provides the platform for the formation of protein during translation. So this is wrong. They are made up of 2 subunits, smaller, larger, that's right. They form polysome. What is polysome? Whenever mRNA, on an mRNA, several ribosomes are attached and making the polypeptide chain. So this is a polysome, single mRNA having multiple ribosomes forming multiple polypeptide chain. They may attach to mRNA, mRNA से तो attach होते ही है, otherwise how protein will be formed. They have no role in protein synthesis. This is wrong. This D is wrong. They do have role in protein synthesis. Next question. Which of the following is not true for a eukaryotic cell? Which of the following is not true for a eukaryotic cell? अच्छा, आपको 6 घंटा हो गया पढ़ते हुए, बस थोड़ा सा देर और W, just 30 minutes more, okay? Just 30 minutes more. Then we will be done. Promise. Just 30 minutes more. Then you can relax, okay? Cell division कर लेते हैं, उसके बाद relax कर लेना. Okay, W? So what do you think is the right answer to this question? Come on, Princey, Shweta, Aruna, W, Neha. What do you think is the right answer? What do you think is the right answer? Good, that's the spirit, my dear. That's the spirit, W. So yes, that's right. Cell wall is made up of peptidoglycans. This is a character. This is a character of a prokaryote. Not a eukaryote. Eukaryote का ये character नहीं है. This is not a character of a eukaryote. 80 type of ribosomes are present in the cytoplasm. That's right. Mitochondria contain a circular DNA. That's right. Membrane-bound cell organelles are present like Golgi complex, endoplasmic reticulum, lysosomes, etc. That's right. So yes, as I told you, everybody, we are done with all the important questions from cell, the unit of life. Now let us solve extremely important must-do question from cell cycle and cell division. I assure you, at least some of the questions that will appear in your NEET 2020 from this chapter will going to be either directly or influenced by the questions that we will be discussing in this class. Remember, NTA has the history of repeating question. They re-repeat the questions they have already given by changing the pattern, changing the sentence, changing the style of asking. But concept वही रहता है, question सेम रहता है, कॉन्सेप्ट वही रहता है, बस स्टेटमेंट बदल देते हैं, क्वेश्चन पूछने का तरीका बदल देते हैं, आंसर को क्वेश्चन बना देते हैं, क्वेश्चन को आंसर बना देते हैं. सो दीज आर वेरी इंपॉर्टेंट क्वेश्चन, मस्ट डू क्वेश्चंस इन सेल साइकिल एंड सेल डिवीजन. चलिए स्टार्ट करते हैं. नेक्स्ट, पहला क्वेश्चन, वेरी इंपॉर्टेंट क्वेश्चन. ये प्रीवियस ईयर क्वेश्चन है और इसके बाद वाला क्वेश्चन भी. दीज आर द मोस्ट कॉन्ट्रोवर्शियल क्वेश्चंस. जब ये पूछे गए थे, मोस्ट ऑफ द स्टूडेंट्स वर in a dilemma, what is the right answer? So let me help you. What is the right answer? Some dividing cells exit the cell cycle and enter the vegetative inactive stage known as G0 or quiescent stage. This process occurs at the end of कौन से stage के बाद में, at the end or towards the end of which stage a cell will enter into a G0 stage? Come on everybody. So that's right, it is G1 phase. So whenever a cell do not want to divide further, they will suspend the cell cycle by exiting the G1. I have given you answer to the next question as well in this one, right? So that's the right one. Now let's see how many of you remember this. Cells in G0 phase terminate the cell cycle, exit the cell cycle, enter the cell cycle or suspend the cell cycle. Come on everybody. What do you think is the right answer to this question? What do you think is the right answer to this question? Everybody give it a try. So I told you, you will be trapped. So इसका सही आंसर क्या है? Suspend the cell cycle. See kids, now listen this very carefully. No, now listen this very carefully. A cell who do not want to continue dividing for some time, they will enter into a G0 stage at the end of G1. So the cells that do not want to continue dividing, they need to exit the G1 at the end of G1 in order to suspend the cell cycle. If a cell is going to exit the cell cycle, it will never be able to return back. It won't be able to divide again, right? So the cells who are entering the G0 are the one who are not going to divide for some time, but whenever they will be called on to divide, they will re-enter the cell cycle. So please remember, a cell which do not want to continue for some time but can again divide whenever it is required, such type of a cell need to just suspend the cell cycle by exiting the G1. Is this clear? So if a cell is going to exit the cell cycle, it can't divide again. हाँ, that's right W, you are absolutely right. बहुत सारे बुक्स में आंसर एग्जिट द सेल साइकिल या टर्मिनेट द सेल साइकिल लिखा है, बट वो सही नहीं है. क्यों नहीं है? आप एनसीईआरटी का किताब खोलो, ओपन द एनसीईआरटी, and they have clearly mentioned that a cell who needs to enter G0 need to exit the G1 in order to suspend the cell cycle. ये क्लियरली मेंशन है. एग्जिटिंग द सेल साइकिल तो कहीं मेंशन ही नहीं है. जो सेल G0 में जा रही है उसे सेल साइकिल को एग्जिट करना है, ये एनसीईआरटी ने कहीं नहीं लिखा है. सो डू नॉट बिलीव ऑन एनीवन, जस्ट बिलीव ऑन योर एनसीईआरटी, रीड द एनसीईआरटी लाइन क्लियरली एंड यू विल बी सैटिस्फाइड विद दिस आंसर, आई अश्योर यू. इज दैट ओके डब्ल्यू? नहीं, अभी आपको किसी का नहीं सुनना है. आप सिर्फ एनसीईआरटी खोलो और उसे पढ़ो. वो सेक्शन पढ़ो जहां पे G0 वाले का डिस्कशन है. द सेल्स दैट एंटर द G0 स्टेज, दे विल सस्पेंड द सेल साइकिल और एग्जिट द सेल साइकिल. क्या लिखा है ठीक से पढ़ो उसे. आंसर आपकी किताब में है, आपको किसी से पूछने की जरूरत नहीं है. इफ यू डू नॉट बिलीव मी, दैट्स ओके. You believe your textbook, and see, your textbook and my explanation is going to resonate because I've also worked on this question a lot, so don't worry. Is this clear? Is this clear W? So let's move to the next question then. Now match again. So let me see who can match this one correctly. Yeah, you can see that later. So try this question, my dear. Match with list 1 and list 2. Come on everybody, give it a try. So S phase is linked with DNA replication. G2 phase is linked with protein synthesis. Quiescent stage is inactive stage. G1 is interval between mitosis M phase and initiation of DNA replication. Is this clear? Is this much clear, everybody in the class? Clear? Can we move to the next question? Yes, I provided the answer over here. Next, mitotic anaphase differ from metaphase in possessing. That's a good and conceptual question. Let me see who can crack this one. Mitotic anaphase differ from metaphase in possessing. Try this question, everybody. Try this question, everybody. what is this? कोशिश कीजिए इस क्वेश्चन का आंसर देने का. Yes? Okay, that's right, W. That's right, Aruna, Princy, that's absolutely right. Now let me help you out in understanding this question. तो है क्या? This is metaphase. This is metaphase. So in metaphase, can you tell me how many chromosomes are there? Chromosome का number तो 3 है. What is the number of chromatids? Chromatids. So remember, each chromosome has 2 chromatids known as sister chromatids. So if there are 3 chromosomes, there will be 6 chromatids. Now this is a scene in anaphase. ये है anaphase. So in anaphase, if you see, chromosome का number कितना है? 1, 2, 3, 4, 5, 6. Because of splitting of the centromere, sister chromatids separate out to form daughter chromosome. So number of chromosome increases, but if you see number of chromatids, it will remain same. इधर भी 6 है, इधर भी 6 ही रहेंगे. Chromosome का number बढ़ जाता है because of the splitting of centromere. So is this clear? Double the number of chromosome and same number of chromatid. Is this question clear, everybody? Next, good. So ये question कहाँ गया? अच्छा, question इस form में है. So identify the meiotic stage in which homologous chromosomes separate while the sister chromatid remain associated at the centromere. Come on. W, Aruna, Princy, Shweta, Neha, try करो इस question को. Everybody try this question. And I'll have a sip of water. Come on, try this question. That's absolutely correct. Very good. So the answer is anaphase 1. That's absolutely correct. So remember, in meiosis, there are 2 parts, right? Meiosis 1 and meiosis 2. In meiosis 1, there will be anaphase 1. और भी बहुत सारी stages होंगी, but I am focusing about anaphase 1. So what will happen in anaphase 1? Homologous chromosomes will finally separate out. And what will happen in meiosis 2? Meiosis 2 will also have anaphase, but this will be anaphase 2. What will happen? What is the characteristic feature of anaphase 2? Here in anaphase 2, my dear students, there will be splitting of centromere. Splitting of centromere. So splitting of centromere is seen in meiosis 2, anaphase 2. In anaphase 2, centromere will split, sister chromatid will separate out to form daughter chromosome. In anaphase 1 of meiosis 1, homologous chromosome will separate out, but sister chromatid will still remain attached. Centromere will not split in anaphase 1. Clear, everybody? Hello Ramlal, yes, absolutely you are late, but this was an extremely important session Ramlal for your coming NEET 2022. So I request you to go through at least the recording or at least go through the PDF. W, is your doubt resolved? W, is your doubt resolved? Yes, okay. Next question. The stage during which separation of paired homologous chromosome begin. अच्छा देखना बच्चों, एक चीज देखना आप लोग, है ना, यहां मैंने क्या लिखा, I was very cautious while writing this. Homologous chromosome finally separate होते हैं एनाफेज 1 में. Everybody, homologous chromosomes finally separate होते हैं एनाफेज 1 में. But the question is, when does this separation begins? When does this separation begins? ये separation शुरू कहाँ होता है? So the stage during which homologous chromosomes separate, starts to separate, begins to separate. Which is the stage? Come on everybody, that's a good question. Come on everybody, that's a very good question. Come on. That's absolutely correct. It is diplotin. तो देखो बच्चों, listen this very carefully. Homologous chromosomes will come together and start pairing in zygotine stage. There will be formation of synoptimal complex between them. There will be formation of bivalents or tetrads in zygotine, which will be even more clearly visible in the pakaitin, right? So two homologous chromosomes, when they come together, pair together, which is known as synapses, right, with the help of a synoptimal complex. This complex is known as bivalent or a tetrad. In pakaitin, there will be crossing over between non-sister chromatids of the homologous chromosomes, which will lead to recombination of the genetic material on the chromosomes. Finally, in diplotin, what will happen? There will be dissolution. There will be dissolution. There will be dissolution of the synoptimal complex. The synoptimal complex that was formed during zygotine will dissolve in diplotin, and the homologous chromosomes will start separating out. But remember my dear students, this separation actually finishes. Finally, actually they are separated in anaphase 1, but separation शुरू हो जाता है diplotin में. Is this clear everybody? मैम सी नहीं होना चाहिए. अच्छा, सी और बी में क्या फर्क है देखना रामलाल. होमोलोगस क्रोमोसोम्स के बीच में जब क्रॉसिंग ओवर होती है, द साइट्स वेयर क्रॉसिंग ओवर टुक प्लेस, द होमोलोगस क्रोमोसोम विल स्टे कनेक्टेड एट दो साइट्स, राइट. सेपरेशन शुरू हो चुका है, बट स्टिल डिप्लोटीन में सेपरेशन शुरू हो चुका है, बट स्टिल द होमोलोगस क्रोमोसोम विल remain attached to each other at the site where crossing over took place. अब इन sites को आप क्या कहेंगे? Chiasmata, right? So in diakinesis, which is a just next stage to diplotin, my dear students, these chiasmata will get terminalized. That means chiasmata will move towards the pole. भाई पोल तक जाना है और धीरे धीरे खत्म हो जाना है ताकि फाइनली होमोलोगस क्रोमोसोम सेपरेट हो जाए. So terminalization of chiasmata is going to take place in diakinesis. Is this clear Ramlal? The difference between diplotin and diakinesis. So diakinesis में और ज्यादा दूर जा रहे हैं वो कनेक्ट होने वाले पॉइंट्स जिससे होमोलोगस क्रोमोसोम आपस में अटैच थे. Is this clear Ramlal? Okay, so can we take the next question then? Aruna, Shweta, W, Princy. Okay, next question. मैच कीजिए. अच्छा, ये सेल द यूनिट ऑफ लाइफ और सेल डिवीजन से मैच द फॉलोइंग के क्वेश्चंस काफी आते हैं. सो बी इन दैट Zone, be comfortable to solve many match the columns from these two chapters. तो बताइए इसमें कौन सी स्टेज के साथ किसका कौन सा फंक्शन कह सकते हैं या कौन सा इवेंट एसोसिएटेड है, बताइए एवरीबॉडी इन द क्लास, कम ऑन. ओके, सो पकाइटीन में क्या होगा या पैकेटीन में क्या होगा? पैकेटीन में क्रॉसिंग ओवर, राइट. मेटाफेज में क्या होगा? ओके, आई थिंक मैंने कहीं प्रेस कर दिया क्या? यस, आई प्रेस्ड आउट. हाँ, सो वी नीड टू गो अ लॉट नाउ. सो सॉरी फॉर दिस वन, बट अब हमें बहुत आगे जाना पड़ेगा. ओके 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 ओके, जस्ट गिव मी अ मिनट. ये क्वेश्चन था हमारा. तो देखना बच्चों, मेटाफेज में क्या होगा? मेटाफेज में क्रोमोसोम अलाइन देमसेल्व्स ऑन द इक्वेटर. मेटाफेज में क्रोमोसोम्स अलाइन देमसेल्व्स ऑन द इक्वेटर. नेक्स्ट, डायाकाइनेसिस में टर्मिनलाइजेशन ऑफ क्यास्मेटा. जाइगोटीन में पेयरिंग ऑफ द होमोलोगस क्रोमोसोम. सो ए गोज विथ 3, बी गोज विथ 3, बी गोज विथ 4, D goes with 2 and C goes with 2 and D goes with 1. So A is the right one. Yes. Next, meiosis results in diploid organism. So एक diploid organisms के अंदर meiosis से क्या होता है? Come on everybody. वीरेंद्र, सॉरी, श्वेता, W, Aruna, Princey, Ramlal, Neha, give it a try, my kids. That's absolutely correct. All are right. Production होता है gametes का. Reduction in number of chromosome because gametes are haploid in number of chromosome means half the number of chromosome and half the amount of DNA. And variation. पैकेटिन स्टेज के अंदर जो क्रॉसिंग ओवर हो रहा है, इट लीड्स टू रिकॉम्बिनेशन ऑफ द जेनेटिक मटेरियल ऑन द क्रोमोसोम एंड हेंस लीडिंग टू वेरिएशन. सो गैमेट्स का जेनेटिक कॉन्स्टिट्यूशन एग्जैक्ट कॉपी नहीं होता है डिप्लॉयड पेरेंटल सेल का बिकॉज ऑफ द वेरिएशन दैट कम्स ड्यू टू क्रॉसिंग ओवर टेकिंग प्लेस इन द पैकेटिन स्टेज ऑफ मियोसिस 1 प्रोफेज 1. सो ऑल आर करेक्ट. सो आंसर क्या है इसका? डी, ऑल आर करेक्ट. नेक्स्ट क्वेश्चन, just few more questions and we will be done. Just for 10 more minutes, my dear students, and these questions will be done. So do not lose the momentum. Keep the josh high. Do not lose the momentum. Keep the josh high. Okay. Next question. At which stage of meiosis does genetic constitution of gamete is finally decided? That's a beautiful question. In which stage of meiosis does the genetic constitution of gamete is finally decided? This is your NCERT exemplar question. Let me see. Please send today's PDF as well. Yeah, so PDF of botany session 1 is already in the Telegram channel and today's also will be given, don't worry. So let me tell you the correct answer is actually anaphase 2. But anaphase 1 is not entirely wrong. Now listen this how. Genetic constitution of gamete is decided in 3 stages. It is decided in prophase 1 when crossing over is taking place, right? Next, it is also decided in anaphase 1 when segregation is taking place. Homologous chromosomes are getting separated, and it is also decided in anaphase 2. So in anaphase 2, finally which chromosome will go in which gamete is done. So determination of genetic constitution finally which chromosomes will be a part of which gamete takes place at the end of anaphase 2. But remember, prophase 1, anaphase 1, anaphase 2 all decides the genetic constitution, but the final determinant is anaphase 2. Is this clear now? So if anaphase 1, anaphase 2 and prophase 1 all are there in the option, you will give the answer anaphase 2. It depends upon the question. In the question itself they have mentioned finally कब decide होगा? भाई finally तो gamete anaphase 2 के बाद ही बनेंगे. anaphase 1 के बाद में तो आपको डायाड मिलता है, टेट्राड तो नहीं मिलता. anaphase 2 के बाद में टेट्राड मिलता है. Is this clear? रामलाल, श्वेता, प्रिंसी, अरुणा. Is this clear, my lovely kids? Can we go to the next question? That's a good question. Please remember this. This is NCERT exemplar question. Next, meiosis occurs in organisms during. 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 Come on everybody, that's a pretty simple question and tricky as well. Meiosis occurs in organisms during. That's right, it occurs during sexual reproduction because sexual reproduction involves fusion of gametes, and gametes are produced. Gametes are haploid, gametes are produced through meiosis. So only sexual reproduction में ही होगा, vegetative में नहीं. In vegetative reproduction gametes do not participate. Okay? Gamete के fusion से नहीं बनता है progeny. Next question in vegetative reproduction. During anaphase 1 of meiosis, what? Now finally I hope everybody will be able to give right answer to this question. Come on. During anaphase 1 of the meiosis, what happens? During anaphase 1 of the meiosis, what happens? Come on now. अब ये आंसर तो सबको सही देना होगा इस बार. Yes, homologous chromosome separates out. That's it. Next question. Mitosis is characterized by. Mitosis is characterized by. Come on everybody in the class. Mitosis is characterized by. Mitosis is characterized by. That's meiosis नहीं लिखा है, mitosis लिखा है. Mitosis. Princey, Shweta, W. This is Aruna. Mitosis. Mitosis is characterized by equal division. Mitosis is equational division. Meiosis is reductional division. ये reduction division meiosis के लिए होता है. Equal division mitosis के लिए होता है. Clear? Is this question clear, my kids? I think you people got trapped in the language, I guess. तो ऐसे जल्दबाजी में question मत पढ़ो. Is this question clear, everyone? Next. A bivalent of meiosis 1 consists of. So bivalent कहते किसको हैं? ये है आपका एक bivalent. और इसको टेट्राड भी कहते हैं. It is also known as tetrad. So इस bivalent में क्या होता है? 2 centromeres and 4 chromatids. How many chromatids? 4 chromatids and 2 centromere. Now listen my dear students, बहुत ध्यान से सुनना, ये वाला concept भी ध्यान रख लो. These 2 are sister chromatids. ये दोनों sister chromatids हैं. ये दोनों sister chromatids हैं और ये दोनों non sister chromatids हैं. ये दोनों sorry, slip of tongue. वो भी sister chromatids ही हैं. ये भी sister chromatids ही हैं. These are sister chromatids. These 2 are also sister chromatids. But these 2 are non-sister chromatid. Now the question is, how will you define sister and non-sister? So remember, sister chromatids same centromere से attached होती हैं. Sister chromatids same centromere से attached होती हैं और non-sister chromatids are attached to two different centromere. Crossing over takes place. I repeat myself. Crossing over takes place between non-sister chromatids. Crossing over non-sister chromatids में होती हैं. Crossing over takes place between non-sister chromatids during Pachytene stage. Is this much clear, everyone? Next. So 4 chromatids and 2 centromere. Cells which are not dividing are likely to be at. Cells which are not dividing are likely to be present at. Cells which are not dividing are likely to be present at. Come on, everybody. The cells which are not dividing are likely to be present at which stage? Yes, which stage? G0 stage, right? Which of the events listed below is not observed during mitosis? During mitosis इसमें से ऐसा कौन सा स्टेज है जो नहीं होता है? During mitosis इसमें से ऐसा कौन सा इवेंट है जो नहीं होता है? Come on. Come on, everybody, give it a try, my lovely kids. Yes, that's right. It is crossing over. Crossing over is an event seen in meiosis. ये meiosis का event है. So chromatin condensation होता है. Prophase 1 में chromosomes बनते हैं. Movement of centriole to opposite pole ये भी होता है. Appearance of the chromosomes with 2 chromatids joined ये भी होगा. S phase में DNA double हुआ है. Next. Identify the wrong statement about meiosis. Meiosis के बारे में कौन सा statement गलत है? Identify the wrong statement about meiosis. Identify the wrong statement about meiosis. Yes, my lovely kids. Come on. Aruna, Shweta, W, Ramlal, Princey. Give it a try. Give it a try, my lovely kids. What do you think is the right answer? What do you think is the right answer? So the right answer, I mean to say, इस question के according incorrect statement क्या है? 2 cycles of DNA replication never takes place. No 2 cycles. DNA replication takes place only once during meiosis, that too before meiosis 1. Right before prophase 1, DNA will replicate. There won't be 2 cycles of DNA replication, but there will be twice karyokinesis and cytokinesis, each in meiosis 1 and meiosis 2. That's right. Next, select the correct statement about G1. Select the correct statement about G1. Come on. Select the correct statement about G1. Select the correct statement about G1. Select the correct statement about G1. Come on, Shweta. Okay, so cell is metabolically inactive. नहीं, cell metabolically active होती है. बायोमोलिक्यूल सिंथेसाइज होते रहते हैं. मेटाबॉलिकली एक्टिव रहती है वो. DNA in the cell does not replicate during G1 phase. ये सही है because DNA replication तो S phase में होता है. This is right. DNA replication does not take place in G1. This B option is right. Is this clear, everyone? Is this question clear, everyone? Come on, let me know in the chat box. Is this question clear? Is this question clear? That's all. We have come to the end of the session. Yes, so you have to maintain that energy. You have to balance yourself. हाँ, तो ये क्वेश्चन तो करेक्ट ही पूछा था, तो ये क्वेश्चन पूछने का तरीका अलग है डब्लू, इस क्वेश्चन को पूछने का तरीका अलग है। करेक्ट बताना है जीवन के बारे में, पर उन्होंने क्या बोला है, डीएनए इन द जीवन डज नॉट रेप्लिकेट, तो ये जीवन के बारे में तो सही है ना। तुम फिर से पढ़ लो इस क्वेश्चन को, देखो दिस इज अ टाइप, इस तरीके से भी क्वेश्चन पूछे जाते हैं। तो जीवन में क्या नहीं हो रहा है, एस अगर यहाँ पे लिखा होता कि डीएनए रेप्लिकेट्स इन जीवन फेज, तो वो इनकरेक्ट होता ना जीवन के लिए। ये बात सही तो है जीवन के लिए कि उसमें डीएनए रेप्लिकेट नहीं होता, डीएनए रेप्लिकेशन डज नॉट टेक प्लेस इन जीवन फेज। ये स्टेटमेंट सही है या गलत है, बताओ डब्ल्यू। सो दिस इज हाउ एनटीए कंफ्यूजेस यू, ये आपको कंफ्यूज करने जैसे क्वेश्चन इसलिए तो बनाए जाते हैं। देखो एग्जाम आपको सेलेक्ट करने के लिए नहीं होता है, रिजेक्ट करने के लिए होता है। ये तो आपको इतना कॉन्शियस रहना है कि आपको सही आंसर देना है, ट्रैप नहीं होना है। एग्जामिनर तो ऐसे ही बताएगा, नॉट अंडरस्टूड। देखो यू जस्ट टेल मी डीएनए इन द सेल डज नॉट रेप्लिकेट इन G1, ये स्टेटमेंट सही है या, अच्छा ठीक है, डीएनए इन द सेल does not replicate in G1 phase। अब बताओ ये स्टेटमेंट राइट है या रॉन्ग है, डब्ल्यू ये बताओ ये स्टेटमेंट राइट है या रॉन्ग है, डीएनए इन द सेल डज नॉट रेप्लिकेट इन G1, ये स्टेटमेंट सही है या गलत है? यस, इज दिस स्टेटमेंट राइट और रॉन्ग? कम ऑन, अभी बताओ ये स्टेटमेंट सही है या गलत है? राइट है, तो यही तो पूछा है, विच सेलेक्ट द राइट स्टेटमेंट फॉर G1। तो दिस इज अनदर वे टू आस्क अ क्वेश्चन, ऐसे भी क्वेश्चन बनते हैं, यही तो समझा रही हूं मैं बेटा अभी तक। So, ऐसे भी क्वेश्चन आते हैं, ऐसे भी क्वेश्चन आ सकते हैं। सो यू हैव टू प्रिपेयर योर माइंड कि आप गलती ना करें। अब ये क्वेश्चन क्लियर है डब्ल्यू? इज दिस क्वेश्चन क्लियर नाउ? इज दिस क्वेश्चन क्लियर नाउ माय डियर? आंसर तो बी ही होगा। ये आंसर तो बी ही होगा, अभी भी समझ में नहीं आया मुझे लगता है। अच्छा ये क्वेश्चन अब क्लियर हुआ या नहीं हुआ ये बताओ डब्ल्यू। व्हाट अबाउट अदर किड्स, बाकी लोगों को भी एक तो डज नॉट लिखा तो है इसमें, डीएनए इन द सेल डज नॉट रेप्लिकेट इन G1। मुझे ऐसा लगता है तुम्हें थोड़ा टाइम लेना चाहिए इस क्वेश्चन को। मे बी हम लोग पिछले 2 घंटे से प्रैक्टिस कर रहे हैं, मे बी दैट इज वाई इट्स नॉट गोइंग। टेक योर टाइम। मैं मंडे को फिर से सेशन लूंगी डब्ल्यू, तुम मुझसे ये क्वेश्चन फिर से पूछ सकते हो, बट आई थिंक आपको कुछ समय के बाद इस क्वेश्चन पे फिर से सोचना चाहिए, आपको ये समझ में आ जाएगा। आप बस इस स्टेटमेंट पे ध्यान रखो, डीएनए रेप्लिकेशन इन द सेल डज नॉट रेप्लिकेट इन G1 फेज, ये स्टेटमेंट सही है या गलत? Right? So I will be taking another session on Monday. If this question is not clear, I can address the same question in the next session as well. Clear? Easy ही था, यही तो बोल रही हूँ मैं इतनी देर से। But such mistakes you don't have to do in the exam, right? चलिए. So thank you very, very much everybody who participated and joined the session as well as became the spectator, although the mute type one. Okay, so thank you everybody, thank you for joining the session and participating as well. So till now, if you haven't joined to Infinity Learn NEET Telegram channel, do consider joining it. The notes, PDFs of this session you will get in the Telegram channel. The link is in the description. So till now, if you haven't subscribed, do subscribe, give me a thumbs up, share with all those friends who are preparing for the NEET 2022, and also hit the bell icon so that whenever I will go live in the next class next time, you will also get notified and won't miss any single session in future. So that's all from this class, my dear students. Thank you very, very much for listening to me. Bye-bye. We'll meet again same time Monday.